My name is Eric Frey, and radiopharmaceutical therapy has meant two things to me. First, it's been an interesting and challenging problem because it's multidisciplinary. And second, it's been interesting because I think it's potentially so promising for the future of cancer therapy.
Hi, Dad. How are you?
I'm doing quite well, thanks. Let's not waste any time. We have the honor and my personal joy in having one of the best, if not the best, quantitative imaging specialist in the field, my friend over many years, Eric Frey.
So how are you doing, George? I haven't seen you in a while.
I'm actually doing well. I'm at physical therapy and occupational therapy. Every Wednesday and every Friday, this is all rehabilitation therapy.
That's great. I just remember the first day that I came in to visit you, not that long after the surgery, you were already talking about your next grant. So I said, okay, well, George is doing okay.
Yeah, that's funny that I mentioned it to you all. I don't remember a lot of what I did when I was in the hospital. So it's a slow-growing tumor. Basically, the approach is to image my brain every several months. I've had this for 30 years. Last time I had surgery and I had some radiotherapy. So the idea is to just monitor it. And if I can get another 25, 30 years, that'd be great. So no treatment until something happens. It's really my left hand and left arm that I can't type with my left hand as fast as I used to be able to.
Hopefully that'll continue to improve. So
yeah, thanks. I mean, the brain healing is really remarkable. And with this rehabilitation therapy, which didn't exist 30 years ago, I mean, you can really recover remarkably quick. So it was kind of funny. I went into the office this week, came in on Tuesday because that's when we have our lab meetings. And I want to focus on getting some of these papers published. I'm at Hopkins. It was group meeting. And when I came out, there were something like 15 to 16 people right outside my office, hanging around, many from the division, the other side of the street, kind of just waiting to see me. It felt like I was at a zoo and they saw me. And it's like when you're at the zoo, is the panda going to come out of its cave kind of thing? Yeah, and all these people are waiting. So yeah, that was kind of funny. Then I walked home. Anyway, Eric will dispense with the formalities. You're Dr. Frey. You've been with Rapid, and you're also professor at Johns Hopkins University. Your specialty is quantitative imaging. One of the advantages of RPT is, and we'll just call it RPT instead of saying radiopharmaceutical therapy each time, is that you can image and see where these agents are going. Imaging is really a nuclear medicine aspect of this treatment approach. But let's first explain what imaging means to begin with when we're talking about these agents. We'll hold off on talking about quantitative imaging and keep in mind that we really need to focus on the basics of what imaging means in this context.
Fundamentally, imaging is a way to try to represent data. And if you think about an optical camera, what you're doing is you're putting on 2D a map of what the colors are in space. And in nuclear medicine, what you're doing is you're trying to make a map, an image of how the radioactive atoms decay and where the radioactive atoms decay in space. And what we do in the kind of imaging that we use for radiopharmaceutical therapy is we try to make a 3D map. So where every point in space in the image represents how many radioactive atoms there were in the patient at that point in space. And that's important because when the atoms decay, then you know how much energy each atom gives. And then that tells you about how much energy is deposited in the patient.
You mentioned for a regular camera, we're detecting colors.
So in a regular camera, you have the photons that are bouncing off the different surfaces, and the light contains a lot of colors in it. And the photons that bounce off the different surfaces have different colors depending on what's absorbed in the surface or not. And in nuclear medicine imaging, what we're seeing is as the atoms decay, they give off photons, they give off light, but it's high energy light, and we usually call it gamma rays. And those gamma rays can travel through the patient and go into the camera that we use to image. And by detecting those gamma rays, those high energy light photons, we get some idea of where the decay happened inside the patient. And so I guess the other thing I would say is when we do the imaging with a camera, you usually take a picture just from one direction. If you just see a flat picture, it doesn't represent as well what we see as people. We have two eyes, and so the two eyes give us some depth perception. So to try to get that in the nuclear medicine images, what we do is we take the gamma camera that can detect these gamma rays, and you rotate that around the patient, and you acquire images of the gamma rays coming out at a bunch of different views or angles around the patient. And then you use all that data to reconstruct with the 3D distribution in space. So from those 2D measurements, kind of conventional images, we use that to reconstruct a 3D volume. And so that's what we're really trying to do in nuclear medicine.
And the 3D set of images you mentioned, reconstruct, those provide the data that you need to really figure out where this agent is going in 3D space, which is neat. You're generating an image of where the decays have happened. Now, we've talked about decays and we've talked about radioactivity. When you talk about radioactivity, people understand half-life. That's what they think of. as a property of radioactive decay. Radioactive decay has two fundamental properties, and I'm sure there are others if you really delve into aspects of decay processes. So one that everybody's familiar with is the half-life, but there's this other one that really leads to imaging, and that's the emission of photons. And in particular, what energy are they emitted at? How does that play into all of this?
The half-life has to do with how fast the atoms are decaying, and then the type of particles that are emitted. So they can emit charged particles like alpha particles or electrons, and those are really important for therapy. But for imaging, we need the light photons. And typically, they emit two kinds of photons. One is gamma rays, which are higher energy. Usually, those have energies of 100 keV or higher, so much higher than light. 100 keV is about the energy of the X-rays that you use when you get an X-ray done at the dentist. The other kind of photons emitted are some of the atoms, when they decay, they actually do emit X-rays, so lower energy photons, typically less than 100 keV. It's very difficult when X-rays have energies lower than 80 keV, they don't get out of the body. So typically the photons that are useful for imaging are in the range of 80 up to, when it starts to get above 500 keV, that becomes hard to image them. It's hard to absorb them hard to measure them in the gamma cameras. So typically the most useful photons are 80 to a couple hundred keV, and sometimes up to maybe even 500 keV. So for PET imaging, the photons that are imaged are 500 keV.
Why can't we see these photons? We can see them on the camera. When you take a picture, you mentioned in both cases they're photons.
In your eyes, the reason you see it is because the photons hit the retina, the cells in the retina, and they're absorbed, and then it deposits energy and then creates neural signals and all that kind of stuff. But the X-rays just fly right through your retina. So in order to see X-rays and gamma rays, you need something that will absorb them. And to do that in the imaging systems, we use crystals that are a material that's dense and it has basically a lot of protons in each atom, because they have a much higher probability of absorbing the photons. And then the second thing we need a substance that'll, either by giving out an electric signal when it absorbs a photon or giving a flash of light, that lets us know when a photon interacted.
I wanted to start with some basics, and now let's talk about some more sciencey kind of stuff. So back to the idea of reconstruction, and you've really made substantial scientific contributions in that area. Can you talk about that?
Sure. Reconstruction refers to using mathematical algorithms to take the projections, the things that we measure in 2D. Basically, if we hold a piece of film up to a patient, the distribution of the gamma rays that comes out in that 2D plane, and we then rotate that, as I mentioned, we rotate that plane around the patient and acquire those projections at a bunch of different projection views. And so what we're trying to do mathematically is to estimate where the decays happen inside the patient that would have produced those set of projections. And one of the keys to getting quantitative images is that the mathematical algorithm you use has to model all the physics that happens when the photons are emitted. So when the photons are emitted, they have multiple energies. And so you need to understand how the different energies affect the images. As the photons travel through the patient, they interact and they can scatter in the patient and change directions and energy, or they can be absorbed. And that's referred to as attenuation, scatter. And those have impacts on the image. And then finally, they interact in the gamma camera itself. Some of the photons scatter inside the gamma cameras or deposit only part of their energies. And so what you want to do is you want to come up with an algorithm that takes into account all those different effects. And then when you do that, then it lets you more accurately reconstruct what happened, where the decays happen inside the patient.
The way I like to explain that is no photon left behind.
I would add to that, no photon left behind and no effect ignored. So you don't want to ignore any of the things that happen to the photons when they're on the way out. The traditional reconstruction methods, when nuclear medicine imaging was first done, ignored the absorption of the photon, the interactions in the bodies, and that results in images that are not at all quantitative. But as we've been able to add modeling for more and more of the physical effects, then the images are a more faithful representation of what's actually going on.
So just to make sure people are following this discussion, we're talking about photons coming from inside the body. We've injected some radioactive material. We've injected RPT. It's distributing. And we've taken pictures at different angles around the body. And we're talking about how we take these individual, basically one perspective images, and using all of them to recreate the three-dimensional distribution of the radioactivity.
Exactly. You can think about, if you're looking at a building, if you look at it from one side, you get one perspective of it. But then if you walk around the building, the other side, you see the other perspective of it. And to get an idea of where the atoms are inside the body, you need to do the same thing.
You've developed a unique, maybe groundbreaking approach to correcting for scatter. Help us understand what you mean by scatter, and then talk a little bit about the technique you've developed that I think a lot of people are using.
I started working on SPECT reconstruction. SPECT is the kind of modality, that the nuclear medicine modality that we're talking about, it's most applicable to RPT. But I started working that in the context of imaging the heart, the myocardial perfusion imaging, the blood flow. And if using the traditional reconstruction method that ignores the interactions in the body, you've got artifacts in the images that look like areas where you had poor blood flow that were basically caused by shadows. The anatomy of different people is different. And so you could have shadows on these images that look like a deep blood flow defect. So we started out working on attenuation compensation. And so I did some work on that. Once you've corrected for attenuation, that means the absorption of the photons. Then the next thing to work on is what happens to the photons that are scattered. And so what scattering means is you have a photon that's emitted from the atom as it decays and it interacts in the patient's body. And when it does, it changes direction. And when it changes direction, it also changes energy. Because it's changed direction, you lose the information about where the atom was. And it turns out that those scattered photons can be detected. And so you have these photons that no longer carry information about the energy. And what they do is they add a fog to the image. So they reduce the contrast. It's if you have an optical camera and you're out on a cold day and you have water vapor that condenses on the lens, the picture you get is foggy. And taking that into account and trying to remove some of that fog is what scatter compensation is about. And I guess the approach that my lab and I have pioneered is to try to model that. One of the things that people have tried to do is try to remove scatter. So they'll take a measurement in another energy window and then subtract that scatter and then use that as the scatter-free images, the reconstruction. But I think this is what you said, George, about no photon left behind. What we've tried to do in our work is to model those scattered photons. So even though they don't contain much information about what's happening inside the patient, they do contain some. And the act of subtracting them is proximate and results in more noise in the images. So what we've tried to do is come up with ways to efficiently model what's happening to the scatter and how it affects those images, and then incorporate that into mathematical reconstruction algorithms so that we can compensate for the scatter by, in a sense, using those scattered photons.
So photons scatter in the body and end up not contributing to the data you like because they look like they're originating from a different place. And that's related to the fog. So what you've developed is a way of taking those photons and using them in a way that contributes to where the original decay was. And I think you mentioned energy window. We haven't really talked about that part of it, which is a more difficult concept. So when these photons come out of the body, they'll have an energy characteristic of the energy resulting from the physical decay. So you can think about energy windows when you're detecting these events. And you want photons that have a lower energy, which means that they're not being detected in the energy window that is consistent with the energy of emission. I don't know how many people we've lost at this point, but we'll try and continue. And if you can contribute to the understanding, that would be great.
And the energy is really analogous to color. So when photons come out of the atom, for a specific atom, they have a specific set of colors. And an energy window is kind of like a color filter. We try to filter out the colors that are not the ones that came from the atom. And that's one way we can get rid of scattered photons. When photons scatter, their energy changes, or in a sense, their color changes. And so we can use a filter to try to filter out those scattered photons. And that's what the energy window is. It's a way to try to filter out scattered photons. And the problem is that the energy windows on the gamma cameras can't measure energy windows with perfect accuracy. So even if a photon has a specific energy, when you measure it in a gamma camera, it'll have a slightly different energy. And so we can't say, I want only photons with an energy of 140 keV. You have to say, I want photons that have energies between, for example, 126, 154 keV. And so when you do that, then you now are accepting some scattered photons. And so that's really why you have a need, even if you use energy windows to project the scattered photons, you still need to model the scatter. And it's an important problem. If you think even a simple radionuclide that's used for imaging, 60% of the photons you measure from cardiac imaging, for example, can be scattered photons. And because each photon is valuable, you'd like to not throw them away. That's really one of the ideas behind modeling them rather than subtracting them.
I really love the color analogies. Let's say you're looking for green, photons that represent green. And as they scatter, they go to a lower energy, and maybe that's blue. I don't know if blue is a lower frequency than green.
Blue is actually higher, but yeah.
Okay. So you're looking, let's say, let me fix it. So you're looking for blue and they scatter and they end up green. But if they're green, they're not really contributing to what you want to get. And you've developed a method that takes the green photons and makes them blue.
I guess the way I would say it is that what we do in the reconstruction is we we try to understand what happens to those blue photons when they become green and where they end up. And by doing that in the reconstructed image, it's not that we take those photons and put them back where they came from, but that we use the color, the new color, to understand where they came from and use that to correct the image in the right way. And I partly say that way because we don't do it photon by photon.
And this is especially when you think of cardiac imaging, this is important for women where in front of the heart, there's breast tissue.
That was one of the initial impetuses for doing that, was a breast shadow. You start doing myocardial perfusion imaging on women, and depending on the size of the breast and how it's positioned in front of the heart, it can give a shadow that can look like a perfusion defect.
And just to define myocardial perfusion, it's not critical to understand radiopharmaceuticals. This is an effort to help people understand what imaging is all about. So tell us about the importance of myocardial perfusion.
Myocardial perfusion imaging, the radioactive drugs you use, the agent that you use is labeled is an agent that flows around in the blood. And what you're trying to see is what areas of the heart have blood in it. And if somebody has heart disease, they can have a blocked artery that means that some parts of the heart don't get blood. And it's important to know if that's actually true, because if a part of the heart that doesn't get blood, then you can put a catheter in there and widen the artery, or you can do a bypass surgery to bypass that part of the artery. So that we're trying to understand is what parts of the heart are getting blood into them.
Let's move to RPTs that are labeled with an alpha emitter. And since alpha emitters are so potent, you can't administer as much as you could in other situations. Like, for example, in the cardiac example, where you can detect where there are enough photons coming out that detection is not a big challenge, except for the need to correct for scatter. So with alpha particle emitters, you're administering a very, very low amount. You're basically barely at the detection level. And most physicists would say, and I know several have, that it's pretty much impossible with so little radioactivity in the body to make them quantitative, to extract from what you measure the number of events or decays that have happened, which is important if you're trying to understand those.
There's an important challenge for imaging the alpha emitters is that there aren't many decays. And the reason that's important is it results in images that are noisy. If you try to take a picture with your camera at night, and cell phone cameras these days have a lot of noise reduction in them. But if you don't have that on, you get a picture that's really, really blotchy. It has some peppery kind of texture to it. That's because there aren't many photons. When there's not much light, there's not many photons. And because in a given period of time, the number of photons emitted is random. And so that results in these random patterns in the images. Exactly the same with alpha emitters. If you take and look at the projections from one position around the patient, there are not many decays. So there aren't many gamma rays that are emitted. And the images themselves will look very peppery.
Gamma rays are basically photons coming from the nucleus.
Exactly. Just high energy photons. They have a color that's way above ultraviolet. Another thing that makes it difficult with the alpha emitters is that the photons have such a high energy, it's hard to image photons that are above 500 keV. So some of the photons for alpha emitters tend to be very high energy, and they scatter inside the patient, inside the camera, and they result in really foggy images. So now you have a peppery image that's layered on top of a lot of fog. What our reconstruction methods do is try to model in very great detail all that stuff that's going on. The scattering in the patient, the scattering in the camera. We use algorithms that take into account the fact that there's that statistical noise, the pepperiness in the images. The other important thing to recognize is we're not just looking at them to see what they look like, and they are going to look noisy. But what we're trying to figure out is how many of the atoms decayed inside the liver or inside a tumor. And by adding up all the regions inside the image for the decays that occurred in those organs, a lot of that noise kind of goes away. It averages out in a sense.
Essentially, incorporated in that approach is the reconstruction scheme, where you take the individual projections and you reconstruct them. And that's where you fold in all the physics of detection. I really would say uniquely, your technique has been able to generate quantitative images that people have just been amazed by.
People used to talk about SPECT as if it was non-quantitative, not even just non-quantitative, but not quantifiable. So they would do ad hoc things, not trying to rigorously correct for things. They would do some kind of calibrations and things like that. And so then when they applied it quantitatively to different patients, they would see really large variations in what they were measuring, maybe a 58% variation in tumor dose across patients that they were estimating from these non-quantitative methods. If you rigorously model all that physics, what we found is that even for difficult radionuclides with high energy photons and not many photons at all, we can get errors of less than 5 or 10% in estimating the number of decays that happen inside an organ. So from greater than 50% by using ad hoc kind of things to 5 or 10% is an order of magnitude of what's achievable.
And when you say model all the physics, I guess it goes back to following a photon from inside the body to outside the body to the detector head and to the crystal. And I guess there's a collimator in between, which kind of means that you're really detecting, let's just say blue instead of red. I'm assuming red is a higher energy. Do I have that right?
Red is lowest energy. Roy G. Biv, ROYGBIV. That's how I remember it. If I know where the photon is emitted, I want to predict what the image looks like. And the physics and the math that let you do that. And once you've predicted what kind of image a photon emitted in a specific place will generate, then you can apply that to the images from the patient. And basically, the patient images is a bunch of photons emitted at a bunch of different places inside the patient. And so by each of those places, you model what happens, you predict what happens, then you can use that to generate an image that is your estimate of what happened based on the estimate of where the activity is inside the patient. And then by comparing that with what you measure, then you can update your estimate, and you use an iterative process. So you repeat that over and over, estimating what the image looks like, comparing it to what you measured, and updating the image. And you repeat that until you get an image whose predicted projections match well to those that you measured. Does that make sense?
Yes. And you've mentioned that when you talk about this process, you're solving matrices that are on the order of 512 by 1024 or some big number simultaneously, which sounds daunting.
I think tomography itself, in X-ray, CAT scans, PET, all those things, we're measuring absorption or emission of photons outside the body, and then using that to reconstruct what's going inside the body. And to me, all that kind of stuff is magic. It's been an incredible advance in medicine. And really some of those ideas, the things that my group has done for SPECT imaging, it all just builds on that stuff done in other modalities and done by other people.
So our producer, Dennis, has suggested that we would pause and take a quick break.
Conversation is going great.
Okay, so we're back, and we sort of have some personal anecdotes. So anybody that's only interested in the science can just sign off at this point if you want to hear some of the more personal stuff, things like journey. The producer is saying, no, you need to stay on. So Eric, do you remember when and where we first met?
Yeah, actually, I do. I'm gonna step back a little bit, give some context. When I came to Hopkins in 2002, Rich Wall got me started on a couple things. One of them was a study of using a high-dose Zeppelin therapy. And I was doing the dosimetry there. You weren't here, so there wasn't anybody to do dosimetry. So Rich asked me to do it. And one of the things that involved is doing blood dosimetry. And so, of course, I came to your paper about blood dosimetry. And so I knew your name from that. So another thing he got me involved in, he was putting together a program project grant to do, I forget exactly what it was, but I was going to run the quantitative imaging core. And just for those who don't know what that is, a program project is a big grant that involves a bunch of different research projects that are all together under one umbrella. And a core is basically a service center that provides some kind of service to all the groups. And so I had written the part about the quantitative imaging. And the day I first met you was when we had the site visit. And the site visit is, they don't do them anymore, but it used to be that you would submit the written part of the grant. And then a few months later, a team of about 5 or maybe 10 reviewers come and they grill you, basically ask you a bunch of questions about problems in the grant and things like that. As I recall, the day of the site visit was your first day at Hopkins. So it was in like March 2003. And they had asked us some questions about dosimetry. And I was working with John Links at the time. And we met with you and tried to come back with a good answer for that. The other place I remember meeting you was at a Christmas party at our house when the producer of this show was much younger than he is now. And we had a white elephant gift exchange. And he wanted to have the bow and arrow set that my son also wanted to have. And so there was some interesting interactions involving that.
Yeah, I remember something about running out in the snow. Dennis got it finally, but it ended up in the basement and never saw the light of day again.
Actually, I think after that, we decided to change the rules of the White Elephant Exchange because while they were good for adults, they weren't so good for younger people.
Yeah. So tell us a bit about the journey. You were at North Carolina for a while and then came to Hopkins.
So I started at Davidson College as a chemistry major, and I really wanted to do biochemistry, but I learned that I really didn't like doing dishes. And when you do chemistry, there's a lot of dishes. So I decided to go into physics where you just had to do equations. I went to University of North Carolina to get my PhD in physics, and I started working on solid state physics in specific related to semiconductor materials. And our group used particle accelerator actually for alpha particles. So we shot alpha particles at silicon wafers and saw what came back. It was a new lab when I got there. So I ended up building a lot of equipment and writing a lot of software. I discovered in that process that I really liked building equipment and writing software more than I did like doing physics. So when I finished my PhD, I looked for a postdoc in medical imaging and ended up accepting one from the University of North Carolina from Ben Choi, who was a relatively young faculty person, and really enjoyed it. I got to use some of the knowledge I had from particle accelerator and physics. And one of the things that's really interesting about medical imaging is it's so multidisciplinary. You have to talk to doctors and you have to talk to statisticians and biologists and physicists, and you're constantly at the edge of not understanding what other people are saying. I think that's been a really challenging and fulfilling part of it.
And how did you get to Hopkins?
Ben Choi's wife was doing population research and she was recruited to come to Hopkins. And Ben had to decide whether to stay at UNC and do a remote commute or go to Hopkins. And Hopkins made a very nice offer to bring Ben and her whole group up there. One of the things I think that's amazing about Hopkins is at UNC, there were a lot of good people, but at Hopkins, there are good people in almost everything. After I came to Hopkins, I got a chance to work with Rich Wall on the dosimetry study. And that was the first time I'd really worked in therapeutic nuclear medicine. And so it was really helped change my career, and having the opportunity to work on those clinical trials and then work with people like you who know the dosimetry part of it is really what's great about Hopkins. You can get people who are good in all the aspects of a project and put together a nice project.
Tell us about Rapid, your thoughts and experience on that. And Rapid is the startup company. I think it's the sponsor of these podcasts.
In 2015, you asked me if I was interested in starting a company to commercialize some of the things we've done. I think we both felt at that time that were things we had done that, you just write papers and then they don't get used, or at least you don't feel like they get used. I had developed methods for doing quantitative reconstruction and they weren't being used. At the same time, I was also looking at a bunch of grants expiring in the next year. So maybe as a backup plan, I thought I'd explore doing Rapid. So I said yes. So we went through the I-Corps process, which is kind of a business boot camp. For those of you who haven't done it, it's a 6-week program where it's really supposed to be about customer discovery. So you're supposed to interview in person 100 potential customers. And first of all, for something like radiopharmaceutical therapy, defining what a customer is kind of hard. Is it the patients? Is it the insurance companies that pay the bills? Is it the physicians who order the treatments? It was a little bit of an inauspicious start. But the I-Corps process was really good. We talked to people that were involved in making RPTs and learned a lot about the difficulties that they have, how they're time sensitive and expensive to get drugs approved. We talked to patients that had been through having a disease that was past any conventional therapies, but had benefits, real benefits from RPTs. We talked to doctors about why some previous RPTs had failed commercially. And so I think just learned a lot, and it helped us to identify what we were going to do in Rapid and to focus on doing dosimetry calculations for drug companies and also developing treatment planning software for the future. And learned a lot to help us launch the company.
So in 2015, we were pretty much ahead of the times, right? I mean, between then and now, and even in just the past couple of years, suddenly billions of dollars are going back and forth. And there are now, maybe 2 to 4 FDA-approved drugs.
Yeah. When we were starting, the Figo had just been approved and had started to create a interest in alpha emitters, and lutetium dotatate had those trials going on. But since those have been successful, it's really created a lot of interest in the field. And as you said, buyouts and a lot of business for Rapid and interest in a lot of other companies making dosimetry products.
You were part of Rapid for quite a while. You were the best amongst us at the finance businessy things. You were the one doing that stuff. And it was pretty clear that you kind of hated doing that stuff.
Yeah. So we actually started Rapid in 2016, and I was kind of the COO, CFO, I think until 2021. I think I stepped down from being the COO. My real goal is to be something like the CSO, the scientific work, and not be involved in the kind of business aspects at all. Partly because I'm 64 years old now, and my wife's a few years older than I am, and I want to enjoy spending more time with her and doing things with her than working all the time as one tends to do in a startup business. I have kind of gone to working part-time with Rapid.
Our producer's asking me to talk more about Rapid, but I'm going to ignore that. what work does Rapid do today that you want clients to know about? So let's skip that question and let's talk about what you did after you left Rapid.
Well, I got a camper van. Dennis is saying, "Arg." I bought a camper van and I spent 11 weeks of summer traveling around the country, seeing national parks and monuments that I hadn't before. I've been to Laos and Thailand in December, and I just got back from spending 3 or 4 weeks traveling around Florida and riding our bikes on a lot of bike trails there. Trying to answer some of the questions that Dennis mentioned about what I think are most proud of at Rapid. As I said, I think I discovered when I was in grad school, I really like to build things. And I think one of the proudest moments I had in my career at Hopkins was some of the software that we wrote in my lab, we licensed to GE for use in their platform to improve the quality of reconstructed images. And when I was at the meeting where they presented that software, it really made me feel good just to know that what I had done was actually being used and have an impact on patients. And I think the same thing for Rapid. I think one of the things I'm proud of is that the first product that we have, 3D-RDS, is a really rigorous implementation of organ-based dosimetry. We put in it uncertainty estimation. We put in it some of the radiobiological dosimetry concepts that you've pioneered, George, and put that in a software product that I think is just easy to use and powerful curve fitting and things like that. The other thing that I'm really proud of is that we do really rigorous dosimetry for people. We know a lot about doing dosimetry well. And so we steer people toward using SPECT imaging rather than plain imaging for dosimetry, using vendor-independent quantitative reconstruction that we've developed that gives consistent performance across different kind of cameras. Especially one of the novel things we've done is imaging of the alpha emitters. For example, we've had customers that have come to us. They want to develop a therapeutic using actinium-225. And one of the difficulties is people thought you couldn't image it. So that means the FDA wants you to do dosimetry to understand where the agent is going. And that means you have to develop a surrogate with an imaging radionuclide. But even when you do that, then you maybe know where the actinium is, but you don't know where the daughters go. So we talked to that company and then showed them we were able to image the actinium and the bismuth and do that directly without a surrogate. And so I think the ability to kind of short circuit that need to develop a surrogate is innovative that Rapid has offered and lets the company bypass that stuff. But more importantly, that the agent will get to the clinic faster because they didn't have that other stuff.
Yeah, absolutely. And we're getting back to the science, but that's fine. Those that moved on after we said we'd talk about the more personal stuff just have lost out. We're getting a note from Dennis into wrapping things up. So with that, I want to thank you very much. This was wonderful. It's a really big contribution to our podcast. We'll have more podcasts like this, really all oriented to helping people understand what this field is all about and raising its profile. Great. Thank you so much, Eric.
Thank you, George. Enjoyed doing it.
All right. How do you think that went?
The early part where we explained medical imaging in the context of nuclear medicine went really well. I thought that would be very helpful to people that hear the word imaging and haven't quite figured out how that works or what it's about. And it's an especially unique and important part when you think of radiopharmaceutical therapy. The idea that perhaps for the first time, you can really see where these therapeutic agents are going in the body and then use that information to figure out how much more they might need or how likely the treatment is to be successful.
I got to say, I wasn't sure if you were getting my feedback in the chat as you were having your conversation. But then when you said, oh, I'm getting these notes from our producer, my son Dennis, my favorite child Dennis, I'm going to ignore these notes. That was helpful because I was like, well, at least he's getting them.
Right. I don't remember saying my favorite child, Dennis.
You did. No, you say it every episode.
Well, you've said it every episode. I didn't.
Well, all in all, Eric Frey was a big deal for our podcast, and I could tell it meant a lot for you to have him on as a guest.
Absolutely. He really is a modest, tremendously accomplished person in his field. I show these images and people congratulate me, and it really should be Eric that's being congratulated. I do my best to let people know that, but because I'm up front and I'm showing the images, that gets lost. People with imaging expertise would know that there's no way that I could do the kind of stuff that he's doing. For the others that are more medically oriented, the message they get is, oh, look, George is generating these amazing images. And it's really Eric and his team. And again, he doesn't feel it's amazing stuff. The background to this is that a lot of physicists just didn't believe you could do this. And Eric delights on being challenged and proving people wrong. So the fact that he and his group have done this is really fantastic. And even though he described reconstruction as magic, it's really just the hard work and using techniques that he's developed over the years putting in the effort to optimize things as much.
Well, I'm not really sure how we should close with that.
Yeah, I'm not either.
Oh, I know. We can direct listeners to go to rapiddosimetry.com and check out the webinar you and Eric hosted in 2023.
That's a good idea. It'll provide a much more technical take on what he's done.
Listeners that want to hear and see more of you and Eric working together, check out the rapiddosimetry.com website and go to the news and resources tab where you can find the webinar.
Absolutely. So with that, we can sign up. Going to the webinar will help a lot. Once this is all together, you definitely want to pass it by Eric because he didn't like these father-son chats.
This has been the Radiopharmaceutical Therapy and Dosimetry podcast with Dr. George Skouras. Produced by me, Dennis Skouras. Executive produced by Dr. Michael Ghali at Rapid Dosimetry. You can rate and review this show on Apple Podcasts. Got a question for George? Or interested in Rapid Dosimetry's products and services? Email us at dskouras@rapiddosimetry.com. And if you haven't already, please subscribe wherever podcasts are downloaded.